Well, this is a real honor for me. And John talked a little bit about Conklin. But, you know, I like to think that, as Shirley said, we stand on the shoulders of giants. And I've always admired Conklin. If you look, it's not just the work that he did in lineage tracing and in embryology, but if you look at the colleagues that he had, and this cohort really formed the roots of modern developmental biology. But for my work, it's really also led me into having an interest in evolution. And when you look at how they really tied the fields of developmental biology and Evo-Devo together, it's an amazing legacy, and I just feel honored to be associated with that. So for me, this is really to celebrate the people. It's a sign that I didn't screw up with all of the talented people that I had working in my lab. But I thought I would take you through, with broad brushstrokes, some of the things we've done as a way of acknowledging people and how we got to where we are. So when I started my lab in London in the UK, we weren't really sure what we were going to do, and it was really an exciting time. Eric and Yanni had done their screen. Ed Lewis had published on the homeodomain. Mike Levine and Matt Scott and others had identified this. And so I was really trying to do differential libraries for teratocarcinoma cells to look for rapidly induced genes, and I cloned Hox genes. And we also had another graduate student who was using homology probes against Drosophila transcription factors. And essentially we got the same thing. And of course I assumed that the graduate students screwed up and mixed all of their clones, but it turned out they independently cloned the same genes from two different components. And they turned out to be this family of transcription factors. And as John told you, I trained as an engineer. And what so captivated me about this is that as we began to look and see that these were ordered, they responded in an ordered way to signaling pathways. They were ordered along the embryo. They had collinearity. It really suggested to me that there was a way, this was an in, to be able to look at the genetic basis of morphogenesis. And I just thought that was so cool. I felt I had to be a part of that, and I've never looked back. So people wonder how I got in and why I've stayed with it. I still don't understand the genetic basis of morphological diversity, but it's going to keep us awake for a lot longer. And another part of that I want to acknowledge, which is the people. So I was an engineer, and Shirley Tillman sort of helped me understand to study genes. We were studying genes in development. But I realized after meeting people like Bridget Hogan and Anne McLaren that I didn't understand development. And that Bridget Hogan took a chance of recruiting me and allowing me to set up my independent lab at Mill Hill. And she was a wonderful mentor, teaching me and exposing me to the joys of embryology and to the members of the community who really adopted me as mentors and taught me an awful lot about embryology. And similarly, at this time, this year marks the 40th year of Ed Lewis's collinearity, his wonderful paper. And again, he was a giant in genetics, and as we began to get more and more data that suggested that maybe the Hox genes in mammals were really true homologs of Drosophila, many in the Drosophila community were not interested or didn't feel that was logical. But Ed, bless his heart, spent huge amounts of time, and Ed was such a scholar, I probably understood less than 5% of the things he was telling me. But the 5% was like gold that have lasted me the rest of my scientific career. So the fact that he didn't have to be interested or have to take the time, and yet he did. And again, Bridget didn't have to do that, but she took the time. And another example here is Denis Duboul. We started out as competitors. We became collaborators, and it feels like we've been lifelong friends. And that's kind of the way my work has gone through in the field, which is why I love developmental biology. But really, again, about the people, I couldn't have been luckier to have my first two graduate students, Nancy Papaloupoulou and Anthony Graham, who are now full professors, and one is a chair of the department in Manchester. They were a bit irreverent. You could see what they thought of me, and we did work with mice here, you can see. But to have people that were so dedicated and so caught into this. And so this has reiterated itself. Wonderful senior mentors. colleagues who have really helped shape our ideas, and fantastic people to make them a reality. And it's just been a fantastic ride, really, basically. So as the lab was trying to think about this and celebrate, of course, working on Hox genes, the Brits put it in temporal collinearity, no order of this, of the people that we have. And then when I moved to the Stowers, there's both spatial and temporal collinearity. There's divided by students and postdocs and others. But as John was saying, I've been lucky enough to have 50 trainees, and now we're up to 28 people that are running their independent labs in 12 countries around the world. 4 people are head of biotech programs, 2 run education outreach. 1 has started a company, Deb Ellis, based on work in my lab, made findings, set up a small business innovation award, and is now chief scientific officer. So really their work in reality pales in comparison to what I've done. So really I want to celebrate those people, and again I thought I would just take you through a few broad brushstrokes and a couple of anecdotes. So while we had these genes, the Hox genes and their collinearity, we were interested in what they would do from a biological system. And this is where, as we began to look, you can see here that the hindbrain, I hope you can see, there's a scanning EM of these undulations or segments. You think you've discovered something new, and then you realize in the archives of the British Library that they were discovered in 1820 in the early heydays of German microscopy. But we showed that these were segmental domains, restricted expression of Hox proteins, mouse mutants would show transformation of them, and really these segments in vertebrates. The hindbrain is a relay center, and it provides a conserved center that enabled us to compare mouse, chick, frog, and fish, to use interspecies comparisons to be able to understand fundamental principles that are not always so easy to do in other regions of the brain. But I think my colleague, my late colleague Rosa Beddington, thought that we were taking the analogy to Drosophila a little too far. And so she made one of my favorite cartoons here saying, we know that the mouse is segmented and we know flies, but make sure you stay grounded. And again, just to show some faces, lifelong colleague David Wilkinson, who has been involved in this, and many other people who were involved in all mapping the gene expression, mouse mutants, and really trying to build up the knowledge for understanding how the hindbrain is segmented. And I think it's the engineer in me that decided if these genes are so conserved, and the toolkit is there, what's different? Are they associated with morphological diversity? I just felt I needed to know the wiring or the circuitry. And so I really started on kind of a very embarrassingly long quest of convincing people that we really needed to understand the gene regulatory network. And I felt that that was important because I thought if we could understand how these genes were controlled, that maybe in different vertebrates, the timing, the levels, or the spatial information might be a basis for evolutionary diversity. So we really made a Herculean effort for that, and it's illustrated here where we can map enhancers or find cis elements, and they came from a variety of genes, and we did regulatory analysis to find factors that are there. And you've heard that from Angela DePace or Emma Fairley, that trying to get enhancers and build stories to be able to understand how signals are integrated is important, but it's a really tough challenge. And what we then did, along with work from people like Cecilia Mowens and Vicki Prince, who using zebrafish mapped many of the early stages of what was happening in the hindbrain, we've come together with kind of a static picture here of what happens at one stage or another stage. And what we learned is retinoids trigger a lot of the patterns of gene expression. Sorry. These genes then mutually cross, repress each other, or auto-regulate, and that then sets up a cascade where they cross-regulate each other, and you then end up with a refined domain. So you can see that this is really complicated. And compared to some of the things that we've been hearing, like eavesdrive 2, where there are some factors that come together that give you a domain, what we began to realize is that we had a big challenge here, and it's illustrated just by this expression of HOXB1 in R4. So Here's the gene and here are a number of cis modules. Here's a retinoic acid response element. And what we know is that if we knock this out in the endogenous locus, we know that somites make retinoids, they diffuse into the neural tube, and that sets up a broad activation through this element. And if you mutate this element, you don't activate them. And then what happens is a repressive element reads directly CROX20, which is induced in this segment and that. An autoregulatory element reinforces expression here. And then what happens is that as development proceeds, the retinoid signal, as somites are laid down, regresses further back. New genes are turned on that repress or sculpt, and so you end up with this precise domain sculpted from a very complex pattern. So you can understand the challenge for us, if we really want to understand this, is the temporally dynamic. When do you turn over the handoff from the initial signal that activates to the maintenance phase, and how is that fine-tuned, and is that the same in all vertebrates? And I'm hoping that this is, again, a really large number of people who have done really fantastic work on that, really trying to dissect this at both at Stowers and at Mill Hill. So where we are now, though, is that this is, as you've heard at these meetings, these sort of cartoons are not very satisfying. They really don't capture the dynamic network. So what we're trying to do is to go back to one species, and we're using zebrafish. And we're using these enhancers and putting tags in the endogenous loci, and really trying to monitor the genuine temporal dynamics to build a full picture in a quantitative way. In a couple of movies, I hope I can show you to see where we're going. Here's an example of the green, or GFP, is the readout of the retinoid gradient from B1, from that enhancer. And we can see the red will be CROX20 coming up. And so I hope you can see that essentially retinoids are spreading up and then they get cleared from this area here. We were surprised that this area is not built, the R4 is not built up a little bit higher, but basically we can follow the temporal dynamics of that. In the next movie, this is an example of how we can look. What I'd like to show you is the dynamic of how upregulation happens in rhombomer 4 and a clearance in this region. So hopefully if you look at this movie, I hope you can see here that you're building up higher and higher levels here. This happens after the repression in these two domains. And then there's also a clearance happening here. And if you look here again, in the next iteration of that, you'll see that it comes up with a small number of cells stochastically. So we can see that this is not really uniform homogeneous expression in these individual segments. And then also another final movie from that. Here's an example of the timing of how do CROX20 and Chrysler work. These are two important early segmentation genes. And it was thought that they came on at the same time, but here we can see in GFP, you will see Chrysler, and you see it coming up. And I hope you can see it's in R5 and in neural crest cells migrating from R5. And here you can see activation of CROX20 in R3, and really stochastic expression in small isolated cells. So this really looks like to us examples of bursting and timing and sharpening. And so we're trying to use single molecule FISH and MS2-MCP to really try to do more detailed imaging to really follow this and quantitate this if we're really going to build a genuine model of how this network works. Okay, and so once we've had that, another phase of the lab, which has been a real, in part, a wonderful collaboration with Marianne Bronner. Hugo Parker in the lab has really been interested in vertebrate evolution, and he's interested in if this hindbrain network is there in jawed vertebrates. Lamprey were thought to not have hindbrain segmentation, and Hugo's done a series of studies where he can show that actually the lamprey hindbrain is segmented. And you can see that Marianne has a wonderful facility, and every summer people come to work at Caltech. Hugo has visited over a large number of years, 6 years, and he's developed tools for making transgenics to do regulatory analysis. And what Hugo's data has really shown is that he's isolated the factors and shown that the regulatory interactions are there in lamprey, and essentially that means that this network was there at the very base of vertebrates. And it's helped us identify ancestral regulatory elements that really give us an idea of how the evolution of this network and cis control have evolved. And that's been a fun thing that is really continuing. And Hugo's also tended that as the lead-in to the next sort of brushstroke, is that I hope you can see here, it's a little bit small. But we then said, well, if it's all the way to the base of vertebrates, what about Siona or Amphioxus? But we have worked hard and we can find no evidence for segmental regulatory elements or origins of those in Siona or in Amphioxus. But what we can find are retinoic acid response elements that are in the same position in the ancient Hox clusters. And that suggests that essentially one way you set up nested domains of Hox expression was through retinoid response, and this might be an ancient feature in chordate lineage. And that's really led us to try to understand more about how retinoid control of Hox clusters work. And just to illustrate a few cartoons about what we think we know about that, again, I told you that somites make retinoids that diffuse in. We know that all of these genes, and for example, this cluster respond, but if we map retinoic acid response elements, not every gene has a response. And so that really suggests, well, how does this work? And Enhancer and other studies in the lab have shown that some of these elements are shared between genes. And it turns out that this is important because you cannot atomize or disrupt these clusters because you can take individual genes and move them apart, but they then lose the ability to respond in cooperative ways with enhancers embedded within them. And so, and part of the way that we've actually done that is we can knock in reporters, fluorescent reporters into BACs or the endogenous loci in a variety of genes and simultaneously in an individual embryo follow expression of all the genes, and then we can mutate elements and show that those expression domains go away. And kind of this final, I think, slide in this area is that what we found is it's more complicated than we thought because we know that long-range regulation of Hox genes is important in heart development. The Hox A and B clusters are important in patterning the heart. And yet these enhancers are only present in placental mammals. And this enhancer is only present in the B cluster, where some of these others are ancient. So it really is looking to us like these Hox clusters can be TADs or topological domains. Enhancers can be evolving within them, and they can regulate, and it's a way of capturing coordinate regulation of a large number of genes. And so we're very interested in the evolution of these long-range cis-regulatory elements. And again, a huge number of people that were involved in this work, James Sharp, Alex Gould, and many others. And I think the final thing I want to say is that our final speaker this evening for the Hamburger Award is Drew Noden. And I talked about what Ed Lewis did for helping me understand collinearity. But when we were starting to understand that Hox genes were expressed in restricted ways in neural crest and were important for patterning, I had the opportunity to meet Drew Noden at Cold Spring Harbor. And over many summers, he patiently explained to a molecular biologist how branchial arches worked. And I had him as a mentor until Paul Traynor, who has been a kind of a recurrent theme as both initially as a colleague and then as a scientific colleague next to me and now as a mentor, has really been involved in much of our neural crest work. And so a lot of what we're doing now is Paul is helping us and guiding us through looking at some of the mechanisms through which Hox genes are coupled to neural crest patterning and how they're functioning in patterning, axial patterning of neural crest. And the final thing I want to notice is that one of the great things about having a lab is that, and such great people, is some people come in and they are just so passionate about something. And you know, we work on Hox genes, and there are these weak moments where I say, God, I'd love to work on anything but Hox. I'm just so tired of it. And we have people like Kenro Kasumi came in and he said, I want to work on spondylocostal dysostosis. And he'd done this beautiful work with Eric Lander. And you just say, God, great person. Let him go have fun. Let him develop independent projects. And so a lot of what we did, I told you about Deb Ellis wanting to work on Wnt modifiers in bone density. I just had a series of people who have done really great work who were passionate, and they sucked me into saying, let's do this and let's have fun. But it can also be serendipity. I've heard a couple of mentions here at the meeting about long-range enhancers from Sonic. And so let me just finish with this last anecdote. This is James Sharp, and he was a graduate student in the lab who really did this beautiful, set up the initial experiments that were showing that enhancers were being shared, or an enhancer was shared between genes. But he had a transgene insertion that had preaxial polydactyly. And he found that much more interesting than anything in the hindbrain. He says, Rob, I just have to finish my thesis on this. So he started working on it, and we positionally cloned it. And it turned out it was in the intron, a megabase away from sonic. And he said, I have to work on this. And he then did his postdoc with Bob Hill, where they characterized this, and this is how they got into the long-range sonic enhancer and the small base pair of mutations that are so far away. And now he's the director in Barcelona of the EMBL unit there, studying limb development. And so you may start out one way, but you can end up, you never know where the serendipity will take you. So again, I just want to thank, I've thanked a lot of my former colleagues. I want to thank the existing lab. I have terrific members who are going on doing things, and my partner in life and my partner in science, Leanne, who was taking this picture, so we had to look for another picture for us. So thank you for your attention, but mostly thank all the wonderful people who have done this work and really made developmental biology a joy for me. Thank you.